From Microbe TV, this is Matters Microbial, a podcast about microbiology, microbiologists, and microbial centrism. This episode was recorded on December 11th, 2023. Hello, Micronauts, and welcome to this week's Quality Quorum. I'm Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 20th episode of Matters Microbial. Can you believe it? I am so grateful to Vincent Racaniello, who sponsored me in this podcast, to the wonderful staff at Microbe TV, to my wonderful guests, and most of all, thanks to each of you as listeners and viewers. I cannot express that enough. First, an amazing and highly relevant member of my Wunderkammer. You may have heard of deep-sea hydrothermal vents, a topic today's wonderful guest will discuss at length. Here is a sample of a hydrothermal vent sent to me, as you can see. Watch the video for just a bit. You can actually see the crystals of sulfur. It's just amazing. I will put several links up in the show notes to these strange and wonderful oases of life. It's also finals week here at the University of Puget Sound. First, a very important reminder to every student, as this button demonstrates, you are each so much more than a grade. Trust me on that. I guess it has been a long semester. One of my Micronauts, students in my microbiology class, made me this swear jar meme about yours truly. Guilty as charged. For all my classes, I enjoy allowing students to do extra credit for creative work. It's my belief that this actually enhances the ability of students to retain and understand difficult concepts. Here's a lapel button made by one of my virology seminar students, as you can see. And here's another button made by a different virology seminar student, as you can see here. And here, remarkably, is a needle-felted model of a virus made by one of my virology seminar students with a key. It's wonderful. And one of my Micronauts, my microbiology students, made me this truly gorgeous stained glass depiction of the three basic shapes of microbes. I treasure each and every one of them. For many years, scientists believed that the depths of the ocean at the very bottom was almost like a desert with little to no life. But in the 1970s, the geochemical vents, often called hydrothermal vents, were shown to be literally oases of life, very far away from the sun far above, instead generating their whole metabolic lifestyle off oxidizing some of the compounds created by these geochemical processes at the vents. Not surprisingly, microbes have found a way to evolved to exploit this wonderfully strange environment. Notice at no point did I say weird. And who better to tell us about these deep sea oases, the symbioses, and his own path in science than Dr. Peter Girguis of Harvard University. Welcome to our Quality Quorum, Pete. It is such a pleasure to have you here today.
Hey, Doc Martin. It is great to see you again. Thank you so much for the invitation to join you and your Quality Quorum here on this amazing podcast, and congrats on 20 episodes. That's so cool.
Oh, it's something I always wanted, and here I'm doing it. So there you go. Do you mind, Pete, if you would give folks, who are listeners and viewers, kind of a thumbnail sketch? I don't know where that expression came from, of your path to science, because you have kind of a background not too dissimilar from my own.
Yeah, happily. So I guess it should start with my parents, who immigrated to the United States from Egypt in the late 1960s, and they moved to California. We found ourselves in Los Angeles around about the time I was 4 or 5 years old, and my mom was an aerospace engineer, and my father is a biochemist, an agricultural biochemist, and they both really came to the United States because they wanted a better life for themselves and their children, and they happened to settle in a town called Downey, which is at the epicenter of the aerospace industry. It was an interesting time in the aerospace industry. It was the mid 1970s. There was a number of houses that were available in this town because the Apollo program, the big moon rocket program, had come to an end. So my parents could find a house they could afford. We moved there, and then the space shuttle program started picking up. And so as a young fellow, I found myself in the midst of this amazing world of building rockets and jet planes and the like. And it was a real heady time. And for me as a kid, that was a time of wonder. I sort of assumed everyone grew up in a little town like this, like with rockets and airplanes and the like. But I was not an especially great student. I was a very anxious kid and dealt a lot with just anxiety in school and subpar performance as a student. And I want to just sum it up by saying that I'm very grateful for those teachers who invested in me. In many cases, sight unseen. I show up in their class, and they took the time to help me through some of my more difficult moments. And so, if we fast forward to college, after years of trying to figure out who I am and what I want to do, I found myself at UCLA, a school that I was really hoping to get into and was lucky enough to get into. There, I started off as a pre-med. I did not like that at all. I bailed on it after a year, focused on Italian history. I wanted to be an underwater archaeologist. And then about 2 years into that, I took an oceanography class and was hooked, if you'll pardon the pun. And from that point, I realized that I wanted to devote my life to studying the ocean in some form or another. I went to graduate school at UC Santa Barbara. There, I worked with an amazing professor who was doing deep sea biology, in particular at hydrothermal vents. And then I found myself at the Monterey Bay Aquarium Research Institute after I earned my PhD, where I was a postdoctoral fellow for about 4 years, again, doing work in the deep ocean. I had no ambitions to end up on the faculty at Harvard or any other prestigious university. I simply wanted a job. I wanted to teach students. That's what I wanted to do, and do research as much as I could. Um, but as the fates would have it, I found myself here, and I've been here at Harvard ever since.
It's funny. I've visited Harvard once, and you weren't there, but I did press my nose against that case that you have at your office. I
Ah,
believe you have do you have a model of Riftia in there?
I do. Yes.
Yes. Yeah, that's what I
Yeah.
did.
Yeah, that case is, I think, probably the most potent recruiting tool that I have. And in as much as I have some specimens in there from the deep sea, some as you saw a 3D printed model of Riftia. I also have some pickled Riftia, as well as some midwater fishes, and our lab mascot, which is a deep sea isopod. And I see right over your shoulder there is a plushy version of a deep sea isopod. We have one that was collected in the Gulf of Mexico, and it's preserved and all, but I call it our lab mascot. So its nickname is Sparkles. And if anyone comes on campus, you're welcome to knock on my door. If I'm not at sea, decent chance I'll be in there. But that display case is there for you all to enjoy and learn a bit about these amazing creatures of the deep.
They're about that size, by the way, friends. Am I not right?
You are spot on.
Hmm,
They're not small.
it's pretty awesome. So I think the next thing, if you wouldn't mind, is to kind of remind people what we mean about a hydrothermal vent and what kind of life you would expect to find at the very bottom. Heck, I remember hearing in high school that there's a thick layer of, I don't know what they call the sediment at the very bottom. There was a name for it when I took oceanography all these years ago. I want to say
Yeah,
almost like Yilm.
I think it was Urschleim.
Okay.
And so, happily, I'll share a little bit about the deep sea. Let me take us back to the end of the 19th century and a time when deep sea exploration was in its infancy. There were a number of expeditions that were towing dredges along the seafloor for the middle to the end of the 19th century. And then in 1872, the HMS Challenger set sail on a circumglobal expedition that would become the frankly the benchmark and the foundation for modern ocean sciences as we know it. That was an amazing expedition. They sailed for 4 years, visited over 360 sampling stations, and they took samples from all around the world, including near the Mariana Trench and the Challenger Deep. They collected some of the first sample from the deepest parts of the ocean. Well, there was a time shortly thereafter that a scientist was looking at the preserved specimens and came to the conclusion that the seafloor must be covered with this primordial early form of life. It looked like a giant amoeba in that there were all sorts of bits and pieces of cells in this viscous goo. And they thought, oh my goodness, this makes sense. This might be the origins of all living things as we know it. And they gave it a name, Bathybius. And it suggested that this was the kind of the sort of predecessor of all life. That wasn't the case. And if you want to learn more about that, there's some amazing books about the Challenger expedition and the discoveries. But let's fast forward to the mid 1970s, about 100 years after the Challenger expedition, at a time when people were just beginning to acknowledge that plate tectonics was real. And plate tectonics refers to the movement of the big plates that we see today, the North American plate, the Pacific plate, and these things that are so familiar to us. But in the late 1960s and early 1970s, this was still not a new idea, but one that finally had some legs to stand on. So in the 1970s, geologists began looking to the deep sea for signs of volcanic activity along what they thought were the margins of these plates. In 1977, a group of geologists came across the first hydrothermal vents on the seafloor off the Galapagos Islands. And this was revolutionary. I would argue the greatest discovery of the 20th century in ocean sciences. Now, what is a hydrothermal vent? When we think of geysers, we think of, say, Old Faithful or other geysers like you might find on land, where you can imagine there's water underground that gets heated. And if it gets hot enough, it comes out through a crack as steam or a water geyser, right? A kind of a gushing water fountain. So we have a kind of intuitive feel for what geysers are because many of us have seen them in film or read about them or learned about them. Now imagine a geyser underwater. It turns out that the world's largest aquifer, where aquifer refers to water underground, the world's largest aquifer is actually the deep seafloor, the basaltic crust. Seawater penetrates through that crust and it moves into zones where it is heated by heat from the mantle. And if you can imagine a kind of pressure cooker where you're down there at a few hundred atmospheres of pressure and the water is being heated and cooked in the presence of rocks, you can imagine that water is going to chemically react with those rocks. And those chemical reactions produce gases and dissolved metals in this hot water, gases like hydrogen, methane, hydrogen sulfide, and others. It also enriches that hot water with dissolved iron and zinc and lead and other metals that are leached from the rocks, if you will. All right, so now you have this hot, high temperature water, and because it's hot, it is less dense than the cold water around it. So it wants to float up. That hot water works its way through cracks in the crust and comes out as a hot spring. And depending on the nature of those cracks and the pressure and the heat and all of that, you end up with different kinds of hydrothermal vents. Some of them look like shimmering diffuse water on the seafloor. Others look like black smokers, giant stovepipes, like a kind of Dickensian, factory smokestack. But that's actually hot water coming out, and it's black because the dissolved metals are precipitating as they hit the ice cold seawater. And so that's the basis of a hydrothermal vent. It's an area of the seafloor where hot chemically altered water enriched in metals, hydrogen sulfide, hydrogen, and other compounds comes out into an ice cold oxygenated ocean.
That is just amazing. I keep wanting to. What's the old? There's a song by Sting about your dark satanic mills, and I wouldn't go quite that far. And I know that many of the hydrothermal vent fields have been given all kinds of fanciful names. There's one off Sweden they've named Asgard, for example.
Right.
That
That's
kind
right.
of thing. Right.
Yep.
So the interesting part is, and I've always told my micronauts that if there's even a slight differential in energy potential, microbes can take advantage of that.
Mm-hmm.
So, for example, it's possible to use hydrogen sulfide as a source of high energy electrons to help drive metabolism. And I believe it's probably pretty true that life is all about an electron looking for a new place to sit. And that's not original with me. It's been said by many people, and it's very true. Now, we're used to that difference that we see dealing with oxygen, but microbes are very clever about this kind of thing. They're very, very cosmopolitan in terms of their electron donors and their electron recipients. So how does that factor into the kind of life you might find around these hydrothermal vents?
Yeah, that is a great question, Dr. Martin. So let's talk about what scientists saw when they first discovered the vents, and then I'm going to loop it back to your question. So, when they first landed on these hydrothermal vents, they saw something unlike anything else they had seen on the seafloor. Most of the seafloor is covered with sediment and fairly sparsely populated. Look, there's life all the way down to the bottom of the Challenger Deep. It's not that the deep sea is devoid of life. It's just in general, most of the deep ocean is sparsely populated because they depend on food from the surface. They're animals like you and me that carry out aerobic respiration. They eat stuff, organic stuff, not organic like organic stores, but organic matter. And they use oxygen to oxidize it. It's kind of like getting energy out of burning a Twinkie, right? Except instead of burning it, you're eating it and you're controlling that reaction and harnessing the energy from it to make a living. That's what you and I do, as well as every animal on Earth, every goldfish, every zebra, every newt, every lion, you name it. We all have this kind of metabolism that's great. You get a lot of free energy out of it, but it means you can do one thing really and one thing only, and that is eat the tissue of other living things and use oxygen to harness energy, right? That's what we do. Now let's pivot to the vents. Instead of seeing a sparsely populated seafloor, they came across these hydrothermal vents that looked like castles that were covered with life, just teeming with life. And I bet you'll have a number of photos that you'll be able to share that illustrate just how extensive the life forms are on these hydrothermal vents. They're covered. They're literally covered. It's like a blanket of living things. Now, how is that possible? Even the geologists, who, by the way, when they first saw this, they're like, well, this is weird, right? They're down there looking for rocks and volcanoes, and they come across giant animals, really vibrant, healthy communities, and they thought, this looks different than what we've seen normally, right? How is that possible? And this is the answer to your question. It turns out that microbes, unlike we animals, right? Microbes have the ability to harness energy from a lot of different chemical reactions. And at a hydrothermal vent, you have hot water coming out that is full of hydrogen sulfide and hydrogen gas and methane and metals. And those are all electron-rich compounds. They've got electrons that are looking for a place to go, if you will. Then they're surrounded by the ocean, just full of oxygen. Absolutely, the bottom of the ocean has plenty of oxygen. It's about maybe half the concentration of what you see on the surface, but it's plenty there. And a lot of the microbes at vents harness energy by taking hydrogen sulfide, for example, that rotten egg smell that comes out of a sewer. That molecule that compound has a lot of potential energy, and they combine it with the oxygen and they harness the energy from that reaction. You and I can't do that. Hydrogen sulfide is toxic to animals, but they're advanced. The microbes harness the energy from these chemical reactions: hydrogen sulfide and oxygen, hydrogen and oxygen, reduced iron and oxygen, and they flourish. There is a lot of potential energy that can be had by taking those reduced chemicals, those electron rich chemicals, and oxidizing them with the oxygen or the nitrate or other similar species. That's it. That's the bottom line. And that is what is the very basis of all life at these hydrothermal vents.
It's also amazing to me that you can get primary productivity based on that. I don't know if many of the listeners and viewers have really thought deeply about this. We always say that the ability to turn carbon dioxide into organic material is something that plants do. And I just shake my head sadly, because the truth of the matter is that's done in chloroplasts, and chloroplasts were at one time bacteria,
Right.
and they have particular sets of enzymes that are able to fix that carbon dioxide into organic material. But doing that is extremely expensive. So here it's run via sunlight because
Right.
sunlight's free. But at these vents, Dr. Girguis,
It comes down to this. What plants do is they harness energy from the sun and they use it to split water, and they use the resulting, let's call them cellular currencies, right, of ATP and NADH and the like. They use that to drive this carbon fixation pathway. It's called the Calvin-Benson cycle, among others, right? But let's stick with the Calvin-Benson-Bassam cycle for now. So here's this carbon fixation pathway. And it is a standalone pathway that if you give it a source of electrons and you give it ATP, you can fix carbon, right? That's what plants do. It's also what algae do and many, many other microbes in the ocean and on land. At hydrothermal vents, in the absence of sunlight, because let's keep in mind that sunlight is essentially gone below 1,000 meters in the ocean. It is never reaching beyond 1,000 meters. Many of these vents are at 2,000 meters, right? Those microbes, they do the exact same thing. And I want to underline that. They carry out the Calvin-Benson-Bassam cycle. That is very similar to what we see in plants. But the energy inputs come from this sulfide oxidation or hydrogen oxidation that I had mentioned earlier. So it is a sunlight independent way of fixing carbon. Now, for the biochemists there, they're going to, I know someone's listening and be like, whoa, 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 time out. The microbes use oxygen. And that is true. That is a photosynthetic byproduct. Okay. So to be clear, if you have a source of electrons and ATP that depends on oxygen, you can drive the Calvin-Benson cycle and fix carbon just like a plant does in the absence of sunlight. There are plenty of microbes that do this without oxygen. Okay, so the punchline is that hydrothermal vents, you have oxygen-dependent and oxygen-independent carbon fixation, or, as you say, primary production. And that is the basis of the food web at hydrothermal vents. And what's really cool about that is that as soon as we saw this in the late 1970s, that you could have these ecosystems that are flourishing, yes, to some degree dependent on oxygen, but to a large degree independent of oxygen, we began to think about the possibility of hydrothermal vents on other worlds beyond Earth, where there isn't enough oxygen to drive photosynthesis. What if there were hydrothermal vents in the outer solar system? Could they possibly have life because there's carbon fixation that can be fueled by oxygen-independent reactions, by oxygen-independent energy reactions? That is so cool.
So, Pete, you were mentioning a little bit about how the kinds of things that you've observed at hydrothermal vents and the way that they drive life could exist on other planets. Perhaps life might have evolved there as well. Is there any thought about the relationship between hydrothermal vents that we've seen on Earth and the evolution of life itself on our planet?
Yeah, that is A really cool question, Mark. And I think let me start by reminding us all that a hydrothermal vent is an environment where different electron-rich compounds are dissolved in water and are coming out into an oxygenated water column, right? It's a underwater hot spring gushing into an oxygenated ocean. Now, it is quite plausible, and if not likely, that there are hydrothermal vents on moons of the outer planets of Jupiter and Saturn. And it is quite plausible that they have many of the same compounds, though not all, but many of the same compounds we see at our hydrothermal vents. And it is quite plausible, again, that they could support some of the kinds of microbes we see living at hydrothermal vents, like methanogenic archaea, for example. That's a microbe that takes hydrogen and carbon dioxide and produces methane. Methane's its waste product, right? That's its version of our CO2. But it takes hydrogen and CO2 and makes this methane. Now, that could happen in a hydrothermal vent in an ocean of an outer icy moon. And when we discovered vents here on Earth, that's when it occurred to us that, oh, wow, this is a lot of life living off of chemical energy. And that was the new idea. See, people had known about this going back to the days of Winogradzki, 100 years before that. But this was the first time we saw this much life teeming around an underwater vent, and began to realize that it's quite plausible you could have an expansive community of microbes living at a vent completely devoid of sunlight and oxygen on another planetary body. That's cool. Now, as we think about the similarities between these vents and what we think are vents on other worlds, it also speaks to the potential origins of life here on Earth, or whether or not it even originated on Earth at all. And what we're beginning to consider now is, where on our planet might there be the right kinds of chemical conditions to support the origin of life? And vents have been a candidate. They kind of go into and out of fashion. Warm springs are a candidate that, again, are currently perhaps more in vogue than vents. But all of these are similar in that we're talking about an environment where there's hydrothermal reactions, water rock heat reactions, right? That produce compounds that might form some of the precursors of life as we know it.
Tell the listeners and viewers the kind of living things that you see there, because I keep running into people that accuse me of being microbially centric, because I am. And then I'll see biofilms and I'll say, look at that life, and they're not impressed. But that's not what we're talking about here.
Yeah, well, life at hydrothermal vents really runs the gamut. We find biofilms. That's an assemblage of microbes that turn themselves into a kind of 1970s yellow shag carpet in many cases at hydrothermal vents. They get that yellow color from the sulfur, right?
Right.
We get microbes that are just like microbes in the rest of the ocean. They're floating around as what we call plankton, right? Free living cells floating around. But something else happened at hydrothermal vents that's amazing, and other similar habitats. Microbes and animals formed partnerships. And what I mean by that is microbes and animals found a way to physiologically work together. Meaning, remember, an animal is stuck with this metabolism of needing organic matter and oxygen. Microbes at vents, many of them, they use the energy from the sulfide, for example, to produce fixed carbon or sugars. And animals and microbes co-evolved to have these symbioses, right? That happened at some time in Earth's past at different times, frankly, among different kinds of animals. But I want to tell you about one in particular, and that is this giant tube worm called Riftia pachyptila. Now, going back to the 1970s, when scientists first came across these hydrothermal vents, one of the things that caught their attention is that there were these worms that they brought up, and they're like a meter long or a meter and a half long, or roughly 3 to 5 feet. These tubes were 3 to 5 feet long, and the worms themselves have the consistency of like a kielbasa sausage. This is not what you normally see in the deep sea. You see things that are gelatinous or jelly-like, right? They don't have a lot of organic matter to make really robust muscles and the like. And then here's this worm. And then it gets weirder. It gets so weird. The worm has no mouth, no digestive tract, no gut, no anus, nothing that looks like it's eating anything. And scientists were flummoxed. They're like, wait a minute, what's happening here? Some scientists said, oh, well, they're probably absorbing organic matter from the water around them. Well, that doesn't make a lot of sense because we didn't measure a lot of dissolved organic matter. Other scientists said, oh, well, maybe they have big old yolks as babies and they just achieve this giant body size because it's warm. Maybe they're just fast. Maybe it's the temperature. Maybe the deep sea animals are flabby and miserable because they're cold. Well, that doesn't make a lot of sense, right? Because at the end of the day, if you don't have a lot of energy, if it's warm or cold, all you're going to do is burn through it faster if it's warm. You're not making energy by being in the warmth. Some scientists even said, oh, maybe they can harness energy from the warmth. And there's just no known mechanism to do that. Two scientists, Dr. Colleen Cavanaugh and Dr. Horst Fellbeck, went and discovered that there were microbes living deep inside the gut of these worms. 10 billion or more microbes per adult worm. That's more than there are people on Earth, folks. And this worm is, in a sense, a giant microbial incubator. And here's the story about Riftia, right? Riftia, as a larva, has a mouth and a gut, and it swims through the ocean, and somehow it finds the right type of microbe. And somehow then it settles and metamorphoses into a closed-off worm and begins to take up hydrogen sulfide and oxygen from the environment and takes it to its symbiont. Now, the symbiont uses the hydrogen sulfide and oxygen to produce energy, and what we call reducing equivalents, NADH, NADPH, you name it. That then drives the symbiont's carbon fixation cycles. Notice I said plural. I'll come back to that in a second. And they produce sugars that we presume are being used by the worm. We don't presume, we know they're being used by the worm. And thus the worm takes up these chemicals that it can't use, but it sends them to its symbionts. The symbionts can use them, and it takes carbon dioxide and produces sugars and feeds itself in the worm. And the worm grows at gangbuster speeds. So that's Riftia pachyptala. It's like the poster child of what we call chemoautotrophic symbioses. That's jargon for chemical self-feeding, chemoautotrophic symbioses. And they are extraordinary animals that I have spent a large part of my professional life studying.
So, Pete, whenever I think about symbiotic associations like this, where there's the large one, the tube worm, and the bacterial symbiont to it, are there relatives of either one of those things that we can learn from to tell us a little bit about how common these symbioses are or how they might have happened?
Absolutely. And the story about the phylogeny, the relationship between the Riftia tube worm and other animals, is a wonderful one. In as much as when we first discovered them, the community gave them their own phylum. They said they're so unique and so unlike anything that they're their own phylum. And it took many scientists, one of whom is Greg Rouse at Scripps Institution of Oceanography, to really start to look at the genetics of these organisms and recognize that they were basically annelids. They're annelids, but they're so morphologically specialized that it was hard to figure that out from their body shapes alone. It really took looking at their genetics to figure that out. The symbiont is the same story. There is a notion for a while that it must be unique, and in fact, no, it has very cosmopolitan relatives among other symbioses. The symbiont in Riftia is not so different than the symbiont in its cousin Regia that lives elsewhere in the world, or Lamellobrachia, another cousin that lives at oil and gas seeps. And neither of those is especially different from a kind of sulfur oxidizing bacteria that just lives in the water column. So these are not so highly specialized, or they're not deeply ancestral. They have commonplace everyday relatives all around us.
I don't know if you're aware of this, but a million years ago, when I lived in San Diego and I was trying to get back to academia, I visited SIO and I met Horst Fellbeck. And he decided it would be very funny to have me help him look at Riftia because he was used to the smell. And he enjoyed my gagging reflex so much when I was there.
I got to tell you, I actually really like the smell. So that is a kind of litmus test, I suppose, as for whether or not someone should, professionally pursue these organisms. They are pungent.
We have a lot of hydrogen sulfide down here, as you know,
I
so we're
do.
going to have to come out here and test you on this one.
Deal.
So I'm going to put a graphic of what a cross section of Riftia looks like, where
Yep.
you have what looks like the top of the lipstick tube,
Yep.
right? And that's the gill that's bringing in stuff.
Right.
And then the majority of the inside is made up of this conglomeration of microbes
Yep.
called a trophosome.
Yep.
And you can actually see, I have anyway, crystals of sulfur in there.
Yep.
Because as they peel those high energy electrons off, what's left behind is elemental sulfur, which to me is just fascinating. The idea of breathing something that makes a solid is just remarkable.
Absolutely.
But the red color is because these worms, people sometimes look at worms and they're a little bit, I guess, homocentric in the fact that they think of us, humans,
Right.
and they'll think that worms are simple. And I suppose you could get away with saying that, but there's a lot of complexity even in a worm. And the fact that worms have hemoglobin, which surprises a lot of people. And I know hydrogen sulfide is toxic to hemoglobin, except for that hemoglobin.
Right. Yeah, really well said. These worms are anything but simple, in my opinion. They have a very well-developed cardiovascular system that they take up the chemicals from the surrounding vent water through that big red fuzzy part called the plume, or which is their gill. That's the really sort of soft, wispy feather duster looking thing that sits on the top of the worm. And so therefore Riftia has to live in what we call diffuse flows, areas where there's hydrothermal vent water mixed with ocean bottom water. And the temperatures that they prefer are like our bathtub temperatures, right? Maybe 20 to 30 degrees Celsius. Warm, right? That's kind of what Riftia thrives in. Now, they take up hydrogen sulfide that comes from the vent water, oxygen that comes from the bottom water, and they move it down through their bodies, down deep into the trunk, into this tissue called a trophosome. And that's what houses the billions of microbial cells. And by the way, those microbes live inside host cells. So the host has built a little cell that they live in, and they almost look like grapes, clusters of grapes. And the tube worm delivers all of those chemicals without poisoning itself. It's astonishing to think that it has these binding factors that bind the hydrogen sulfide, chemically bind it, and take it down to the tube worm symbionts. And here's the part we don't know how it works yet: release that sulfide and that oxygen without causing problems. And the symbionts do their stuff. And then the worm also gets rid of their waste products, which includes proton equivalents, or acidity, if you will. So the worms actually have to pump out protons, or hydrogen ions, to keep their bodies from being poisoned through acidosis or having too much acid. So these worms are phenomenally cool.
Do we know how quickly they grow, or is that very hard to calculate?
We are publishing those data and have published preliminary insights into those data, and they grow about as fast as some of the fastest growing photosynthetic organisms. So they can keep up with kelp forests. They can certainly keep up with wheat, bamboo. And so they are incorporating somewhere north of about 10% of their body carbon a day. Now, for someone of my age and size, I'm sorry to say, that is a terrifying thought. But for these worms, it is a huge advantage in a hydrothermal vent environment. And very briefly, it's advantageous because it means that Riftia can outgrow a lot of the other organisms. It can get to a vent that's newly formed, it can settle, it can grow quickly, produce its gametes, have all of its babies. They basically live hard, live fast, die young. That's the tube worm lifestyle.
So what I was thinking is I was taking the old Beatles song Octopus's Garden, and I'm
Right.
thinking about a hydrothermal garden, but it's really more like James Dean when you come right down to it.
Uh, I'm all ears. How was
No,
that?
it's just well, because live fast, die young. That's the whole
Oh,
thing, right?
I missed my own joke. Yeah, absolutely. It's very James Deanish. It's spot on. That's what they do.
And it's fascinating to me. A couple of things. I mean, I look at this from kind of a bacterial point of view, and I know they don't have a point of view, but to me they do. And one of the problems is finding your macroscopic partner.
Yep.
And a friend of mine from a very long time ago, Debbie Milliken, was able to show that when you take the genome of the Riftia organisms and you
Hmm.
try to see whether or not any of those genes are active, she was able to show that the motility genes were active. So this isn't something that doesn't live outside
Right.
the host. I mean, it obviously undergoes some kind of change, much as rhizobium does in a root nodule,
Yep.
some kind of change so it no longer can live independently. But this has to be part of the dispersal issue, which is fascinating to me.
Yeah, absolutely. And we know that each individual tube worm acquires its symbiont anew from the environment. So every time a Riftia larva is swimming around, it has to find its symbiont. And it seems most plausible that those microbes are living somewhere on or around the basaltic crust down in diffuse fluids, and these larvae find them, somehow ingest them, and then somehow settle. There's a lot of mysteries surrounding this, though. The larvae seem to be buoyant, they seem to float. Or certainly the Riftia gametes, the kind of blastula and other different developmental stages seem to be buoyant. And so they're somehow up in the water column, we think. And if that's the case, how do they come back down and find their symbiont? There's a lot of mysteries that still exist about this association that I think are fascinating.
And one of the things that I hope our listeners and viewers understand is that things that live very far down deep, that's the technical expression for it, don't do well when you bring them all the way up to the investigator.
Right.
And these are deep enough that you need to kind of lift them up if you're going to do that. You can't send well, you can send a submersible down there, but you can't do the kind of work that needs to happen. So just as my fishermen friends would bring up a deep sea fish and it would not do well on the deck, this is the situation with Riftia. And I know that's what I'm leading into, what you did next. Please tell.
Absolutely. As a graduate student, my advisor, a fellow named Jim Childress, was building pressure vessels to keep these worms alive. And what they had done was build these large metal tubes, put worms in them, pump those metal tubes full of high pressure surface seawater, and do the best they could to keep them alive to do their biochemical assessments. As a first year graduate student, when I joined Jim's lab, I said, "What are the project options?" And he said, "Well, lots to do. There's more biochemistry on the hemoglobins. There's looking at carbon fixation. There's all these other things." And then he said, then there's this high pressure respirometry system. Try it for a while. But, I've been trying to get someone to work with it for 10 years to varying degrees of success. The people before me they kind of got it to work for a couple of animals, published a couple of papers. But he said, this is something where if we can get this to work, we can measure their metabolic rates. Okay. So, but I took to it. It tapped into my love of all things mechanical. And I started refining and adapting the system, starting with Jim. I don't know that pressurized seawater is good enough. Can we really recreate the chemical conditions in situ? And he said, well, we've tried that, and it doesn't work. And I said, well, let's try it this other way. Can we try bubbling with sulfide instead of adding sodium sulfide salts? Or can we try controlling the pH? And he's like, oh, yeah, well, yeah, you can try, but I'm warning you, this is hard. Well, I give him credit for letting me try and try and try again. And I went out to sea for about 2 years, spending several months at sea trying to keep tube worms alive. And it was working, but things were not as vigorous as we had anticipated. We could barely measure their metabolic rates. And then one day, 2 years in, the water bath I was using to keep these pressure vessels cool at about 7 degrees C, it died. And the temperature inside the pressure vessels on the East Pacific, right? We're near the equator here. We're about 9 degrees north. The temperature shot up in these pressure vessels to about 23 or 24 degrees Celsius. And my advisor, who was not, was sort of impatient at times, came down swearing and cursing and the like. But then I looked at the data and I said, holy smokes, Jim, these things are going gangbusters. And he goes, impossible. It's the free-living microbes. So I opened the pressure vessel up, took the worm out, put its tube back and sealed it up and said, okay, if it's free-living microbes, we should get the same rates. And we didn't. It was the tube worm. That is an important moment in my career, and it was serendipitous. Because at that point, it occurred to me that we were doing this all wrong. We were being very conservative, cool temperatures, modest pHs. Even though we had data from the seafloor that suggested that they lived in warmer, more acidic environs, we were tiptoeing there. At that point, I'm like, gloves are off. We're going warm. We're going acidic. And the worms loved it. Now, fast forward to today, I've built this intermodal shipping container based on what I learned from Jim in the lab. I've taken an intermodal shipping container, which is a railroad car or a truck car, and turned it into a mobile high-pressure lab. In there, we have high-pressure pumps, high-pressure vessels, and it looks like a kind of space station, if you will. But what we're able to do is pick up this entire box from Harvard University, put it on a truck, get it to a ship, bolt it to the deck of the ship, plumb it so we have access to seawater and gases and all that. And then we use our robot subs or human-occupied vehicles to go down, collect worms, and as quickly as we can, get them in the pressure vessels. At that point, they do great. They're happy. We recreate the conditions that they were living in, and we can do those respirometric metabolic rate measurements better than we've ever been able to do before. And these days, we tie it into transcriptomics and proteomics to look at patterns of gene expression by the symbionts and the host to see how they respond to changes in environmental condition. And I'll end by saying I mentioned 2 carbon fixation pathways, or a plural. Well, that's because the symbionts unusually have 2 ways to fix carbon: the Calvin-Benson cycle and something called reductive TCA. And what's really cool is that they seem to be using both in parallel to some degree, and it seems to be modulated based on the environmental conditions. And we're just getting this paper out now, and I hope that it opens up a whole avenue of exploration for others to run with this story of how an organism uses 2 different pathways to achieve something that we rarely see in other organisms. That is, these extraordinary carbon fixation rates.
This is just an amazing story. And it reminds me of when I was in graduate school and we were studying rhizobia, which form these root nodules on leguminous plants, and they're able to fix atmospheric nitrogen inside there. And there's a whole story behind there. But the important point is when those bacteria are in the plant cells, the plant membranes are restricting what the bacteria perceive and use.
Right.
So even though, for example, easily 40% of the cytosol of root nodule is sucrose, the bacteria don't use sucrose at all.
Right.
They use dicarboxylic acid. So the whole take-home message is that this is a ballet that's been Darwinoed over evolutionary time. They've been really fit together. It reminds me very much of the changes that took place in the bacterium that we now call mitochondria inside every one of our cells.
Right.
They've been fine-tuned to their functioning.
Absolutely.
The difference is we don't have mitochondria living inside of us. They're just transmitted vertically.
Right.
through the eggs. So this type of symbiosis is just fascinating. It's a nice snapshot of what's taking place midway, trying to become an organelle, if you wouldn't mind my saying so.
That's very, very, very good point. And it's a reminder that, and I think this is important, really important for all of us to keep in mind, that our studying organisms is itself a snapshot, right? They're constantly evolving, sometimes faster than we can pay attention to, and often slower than we're able to be aware of. And so we always have to keep in mind that it's like standing, on a crowded parade route where you can only see what is, the parade that's right in front of you. You really have a poor sense of a poor memory of what may have passed 30 minutes ago, and you have no idea what's to come. And so it's important that as we study these organisms, we keep in mind that there's a bigger, more expansive picture, both in the past and in the future. These organisms are headed on a trajectory that we can't always predict. So it's a very fun part of being a biologist, is knowing that everything is constantly changing. So one thing you can
Oh,
count on.
it- it's like, as you may have heard, because I went to UCLA too, so you probably had some of my professors, and I had a physics
Mm,
professor who said that the problem with physics is that variables don't and constants aren't.
yes, exactly. Yeah.
And so I guess that's been with me all these years. And so what I often say to my students is that the hand of Darwin is on all that lives, even in
Hmm.
the process of cultivating in the lab. And that's a really important point. It's so
Yeah.
hard to do that. My own students see this when they isolate, say, a pigmented bacterium from seawater. After a couple of isolation rounds, it loses that pigmentation. It's
Right.
extremely common.
Right. Which is cool.
Yeah, and that's why when you create this specialized compartment where you can study Riftia in situ, and that some of the pictures are just lovely. I'm thinking about all the high pressure behind that and worried about that, but that's just me being a worrywart. But there they are, their plumes out and they're taking things in. It's just a wonderful story, Pete.
Well, I appreciate that a lot. And I got to say that studying these organisms under pressure appeals to my engineering tendencies. And I want to say to your listeners out there that each of us has something to offer the natural sciences. You really do. You can be an accountant and still bring your skills to bear on better understanding our world, or a lawyer, or an engineer, you name it, right? And one of the things that I've loved doing throughout my career is bringing my engineering tendencies. And I am not a card carrying engineer, but I know the questions that need to be answered. And that puts me in a position to think of tools and technologies that can help answer those questions. And it's really been fun to stand in this pressure lab, this mobile pressure van, and have my face within just a few centimeters of this high pressure vessel, and they are very safe. And I will happily explain to you why at another time. It's really, water is not that compressible, right? At the
Yep.
end of the day, when the pressure vessels burst, which has happened maybe once in my career, you end up with a lot of water everywhere. And we take great precautions. I don't want to sound cavalier. Like, we wear our safety gear and we're careful, but it's amazing to think that there, in the presence of this pressure vessel, is an animal that came to us from thousands of meters below the surface of the ocean. This animal that has never seen the light of the sun until it came up on this robot sub or human occupied vehicle, and then we put it back in its dark little home, and it loves it. It's just a reminder that, in many ways, when you think about the fact that the deep sea is 80% of our planet's living space, right? That is normal for our biosphere. Living in deep, dark, high pressure seawater, that's the normal state for our biosphere. It's the majority of our biosphere. So we're the odd ones out, right? That's why I think we're the weird ones, not the worms.
Oh, I agree completely. I mean, you've probably heard me give the talk. People, when they hear microbes, they think of all they do is terrible things or all they do is good things. I call those like devil microbes and angel microbes.
Right.
But the fact is, most microbes are meh. They don't care about us at all.
They really don't. And
No.
that, they're like basically running the world,
Mhm.
and we're along for the ride. That's the punchline, I think.
It's a change off Beyonce's song. I mean, who runs the world? Germs, right? And that's really what it is.
Yep.
Um, so I just want to make clear to everybody that one of the things that makes Pete such a great scientist is that he is what I call, and this is a capital M, a maker. He tends to build things. And I think of this a lot because I don't have a lot of skills in that area. So it's really impressive. And that's part of everything that he does. And it's really remarkable.
You're very kind. Thank you, sir.
And I wanted to ask you a question. I like to ask visitors to the podcast what one thing sold them on microbial science. So, for me, it was the first time that I saw bacterial bioluminescence, that kind of thing.
Right. I think it was the partnership with an animal.
Yeah.
I think for me, it was the idea that what I thought was a really cool animal turned out to be equally as much a really cool microbe. I went to Jim's lab to study the worm, Mark. I didn't go there for the microbes. I actually never even took a microbiology class as an undergraduate, right? It wasn't until grad school that I started learning everything I could about microbes. But I went there to be a comparative animal physiologist.
So you came for the worm, but you stayed for the microbes.
Yeah, you got it. Heck yeah.
That's a wonderful story. Do you have any other bits of wisdom that you'd like to share with listeners and viewers?
I think that it's important to keep in mind that our job as scholars, as humans, as people who are living on this earth, and if we're going to study ocean sciences, our job is to bring the best of ourselves to studying the ocean or the world around us. I mean, literally, it's I think I'm trying to say, just be the best version of yourself. I've spent and wasted a lot of time thinking about trying to be a version of someone else, especially as a graduate student, where I was just so in awe of many of the grad students I worked around. I thought, oh my gosh, I can never be as good as them. I'm going to try to be as good as them. But at the end of the day, I really just need to be the best that I can be. That brings my unique perspective to our collective understanding. And mine is no more important than yours or anyone else's. So that's my 2 cents. Just be the best version of you and don't worry about the rest.
I've heard it over and over again that authenticity really matters. As you are painfully aware, I'm a little different than most scientists. And there are people who like it and people that don't. But I cannot tell you the number of times that I've heard graduate students say to me privately, thank you for being as, I guess the term they like to use is awkward. Thank you for being as awkward as you are, because it's who you are. And I think that it comes across as being really the truth. And that's what people need to see. Scientists aren't magical. It's a job like anybody else has. It's just there's different questions and different approaches. And
Right.
once you're yourself, you're a little bit happier with what you're doing.
100%. Yep.
Well, Pete, thank you so much for visiting the podcast. I was getting a little bit carried away. Perhaps I can have you on another time and we can get into other forms of symbiosis, because I would like to pick your brains on, well, the microbes in your brain on that particular topic at some point. But I really appreciate your time.
The pleasure is mine. Thank you so much for inviting me. And thank you, listeners, for sticking with us.
And my very best wishes to your family.
Much obliged. Same to you.
Thanks, Pete. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes, with tasty links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Pete Girguis can be found in the Department of Organismic and Evolutionary Biology at Harvard University. Many, many thanks to David Renata for superb editing and fine questions, and Reber Clark for the wonderfully quirky music. I hope you've all enjoyed being part of our quality quorum today. See you next week on Matters Microbial.